This bluish liquid is one of the most expensive resources in the world. No, it's not the blue milk from Star Wars. It's actually blood from a horseshoe crab. And the stuff this blood makes costs $60,000 a gallon. So why is it so expensive? And who's buying horseshoe crab blood? The blue color comes from copper in the blood. But that's not its most interesting feature. The blood contains a special clotting agent. It's used to make a concoction called limulus amoebocyte lysate, or LAL. Before LAL, scientists had no easy way of knowing whether a vaccine or medical tool was contaminated with bacteria, like E. coli or Salmonella. Scientists would inject vaccines into huge numbers of rabbits and then basically wait for symptoms to show up. But when LAL was approved for use in 1970, it changed everything. Drop a minuscule amount of it onto a medical device or vaccine, and the LAL will encase any gram-negative bacteria in a jelly cocoon. While it can't kill the bacteria, the jelly seal is like a fire alarm, alerting us to the presence of what could become a potentially lethal infection and prevent it from spreading. Each year, the medical industry catches around 600,000 horseshoe crabs. The crabs are drained of 30% of their blood, and up to 30% of the crabs don't live through the process. The survivors are returned to the water, but no one really knows how well or if they recover. In 2016, the International Union for the Conservation of Nature bumped the American horseshoe crab up to vulnerable on its red list, one step below endangered. And the US population could keep falling by as much as 30% over the next 40 years. LAL labs claim that the returned crabs eventually recover, but new evidence suggests that's not always the case. Wynne Watson is trying to figure out what happens to the crabs when they're put back in the sea.
So the most immediate negative effect is mortality. Anywhere from 10 to 25% of the animals will die within the first couple of days after you bleed them.
Bled crabs become disoriented and weak for a period of time, and females may have trouble spawning.
If they survive the first, I'll say 2 weeks, and they're back in their natural habitat, they did pretty well.
But it's getting through those 2 weeks that's the issue.
Based on our data and others, I think that you need to treat them a little. If you're going to get increase your survival rate, right, you need to treat them better.
Scientists are trying to find a synthetic alternative to help reduce the strain on the horseshoe crab population. But so far, LAL is still required by the FDA for this type of testing. So if these animals really aren't recovering at the rate companies previously thought, we might eventually run out of crabs to bleed. If that happens, our lives, and the lives of countless rabbits, might be at risk.
Every year, hundreds of thousands of horseshoe crabs arrive on the beaches of the Atlantic coast of America to lay their eggs. And every year, hundreds of thousands of horseshoe crabs are rounded up and brought to the lab, not to be killed, but for their blood to be carefully extracted. These animals, often called living fossils, are one of the oldest creatures on the planet. They've remained nearly unchanged since they first appeared on Earth over 450 million years ago. This is due to some exceptionally effective adaptations and genes that code for remarkable molecules that have allowed the horseshoe crab to survive, just as it is, for so long. One of these ancient compounds is the reason that hordes of these animals are dredged up from the ocean, jabbed with a hypodermic needle, and their blue blood drained, processed, and sold. Their blood, made blue from a copper-based oxygen-carrying molecule, is so valuable that it's the basis for a multi-million-dollar pharmaceutical industry. So valuable that a single liter of it goes for around $16,000, one of the most valuable liquids on Earth. Our reliance on these animals puts immense pressure on a fragile ecosystem. And so far, scientists have struggled to recreate this compound and its effects in the lab. What is it about this primitive compound that we need so badly? And why can we only seem to get it from this one creature? The American horseshoe crab is an ancient aquatic arthropod. They belong to their own class of animals, called Merostomata, and are not actually crabs. They're more closely related to scorpions, with their predecessors diverging from their arachnid cousins around 480 million years ago. Some recent studies even suggest they are arachnids. The modern horseshoe crab, as we know it, has technically only been around for 20 million years, but some of its early relatives, like Limulus darwini, existed around 150 million years ago and look nearly indistinguishable from today's horseshoe crabs. The iconic body plan has been around even longer, emerging around 450 million years ago. The changes within the horseshoe crab group have been shockingly minor in the big picture of evolution. Here's some perspective on just how long ago horseshoe crabs came into existence. Pangaea, the supercontinent of the past, formed 335 million years ago and began to break apart about 175 million years ago. The non-avian dinosaurs emerged 245 million years ago and were wiped out 66 million years ago. The Earth descended into and emerged from two completely different ice ages since these crabs came about. The world has changed so much since then, and all the while, horseshoe crabs have been here, crawling along the seafloor, standing the test of time. Some of the reason natural selection has preserved them, pretty much as they are, is their hardy body plan. Their hard shell, called a carapace, is an exoskeleton so strong that only sharks or turtles can penetrate it. And guiding them through the ocean depths are 9 eyes, 2 compound eyes which act much like our own eyes, 5 secondary simple eyes on top of their shell which can detect UV light, and 2 ventral eyes located on their underside, perhaps to help with orientation. Along with their complex circulatory system, 5 pairs of gills, and 12 bristled legs, evolution created them to be creatures extremely well adapted to their particular environmental niche. But beyond the physical traits that we can observe, much of their survival is due to something we can't see: their incredible but simple immune system. It's protected them as a species from bacterial infection for eons. It works in an entirely different way from ours, and in the late 1960s, we began to harness its power for ourselves. In 1968, 2 researchers at the Marine Biological Laboratory in Massachusetts observed that blood cells from horseshoe crabs vigorously clot in the presence of bacterial endotoxin. When they published their paper, they had no idea that what they found would revolutionize drug safety testing forever. Pretty much every creature in the world is vulnerable to bacterial infection, and the horseshoe crab is no exception. Once an infection begins, bacteria can reproduce quickly, and many give off toxins which damage specific tissues in the body. Botulism, for example, is an illness caused by a neurotoxic protein produced by a bacteria called Clostridium botulinum. The toxin can affect your nerves, paralyze you, and even kill you. Toxins like this are called exotoxins. They are released from live bacteria into the surrounding environment during an infection. But bacteria don't have to release these exotoxins in order to be dangerous. In fact, they don't even have to be alive. Once a bacteria is killed within the body, they sometimes release endotoxins. Endotoxins are the lipid portions of lipopolysaccharides that are part of the outer membrane of the cell wall of some bacteria. The endotoxins are released when the bacteria die and the cell wall breaks apart. This toxin is a pyrogen, a fever-causing agent. If it gets into the bloodstream, it can lead to septic shock and can be deadly. But fighting off these types of infections is what immune systems are for. Immune systems have developed to protect all different kinds of organisms from foreign pathogens. And during the course of evolution, 2 different kinds of general immune system emerged within multicellular organisms. Humans and many other vertebrates have adaptive immune systems that protect us by strategically mounting a defense against invading bacteria. It's activated by exposure to pathogens and uses an immune memory to learn about the threat and enhance the immune response accordingly. But many invertebrates, including horseshoe crabs, don't have this adaptive immunity. Instead, they have an innate immune system, which attacks based on the identification of a general threat. The basis for a horseshoe crab's immune response are cells called granular amoebocytes. When bacteria come into contact with a horseshoe crab's blood, they trigger an enzyme cascade, mediated by these amoebocytes, which causes the blood in the immediate area of the infection to clot into a gel. The gel surrounds and isolates the infection from the rest of the crab, and the pathogens are neutralized. The clotting from granular amoebocytes is a simple but very effective way for the horseshoe crab to defend itself from infection. And, as researchers began to realize in the 1960s, it's a very effective way for us to detect the presence of toxins in places where we really don't want them to be. When creating injectable healthcare products like vaccines, medical implants, and IVs, it's imperative that they are free of any invading microbes. It's easy enough to sterilize the solutions or devices by blasting them with heat, radiation, or gas that's deadly to bacteria. But killing bacteria isn't enough to make these products safe. If certain bacteria were present before sterilization, the endotoxin will remain and can lead to severe consequences if injected. Historically, pharmaceutical companies got around this problem with huge colonies of rabbits, needed for what's called the rabbit pyrogen test. To see if a product or drug is contaminated with endotoxin, 3 unlucky rabbits would be injected with a small amount of the drug or product in question and monitored for 4 hours. Rabbits have a similar pyrogen tolerance as humans, so if any develop a fever, the batch would be considered to be contaminated with bacterial endotoxin. This is an effective way of preventing endotoxins from accidentally being injected into the public. But because it's an in vivo test, meaning done inside a living organism, it's very time-consuming and expensive. So when researchers noticed the clotting effect of the horseshoe crab's amoebocytes in the presence of endotoxin, they realized it could be an in vitro way of spotting contamination, a much cheaper, easier, and faster test. This in vitro test is called the LAL test, Limulus amoebocyte lysate, Limulus being Limulus polyphemus, the American horseshoe crab. This test has become the worldwide standard for screening for bacterial contamination. It's capable of detecting endotoxin at significantly lower levels than the rabbit pyrogen test. Today, every drug certified by the FDA must be tested using LAL, as do surgical implants such as pacemakers and prosthetic devices. After the horseshoe crabs are brought to the lab, the tissue around their heart is pierced with a needle, and up to 30% of their blood is drained. The amoebocytes in the blood are then extracted from that for the LAL test. Upon exposure to endotoxins, the amoebocytes undergo a rapid enzyme cascade that causes the cells to stick together and form a clot. This clot can form in around 90 seconds, giving a nearly instant result. We've never found anything that's as sensitive in detecting endotoxin than the horseshoe crab's amoebocytes. If there are dangerous bacterial endotoxins, even at concentrations of 1 part per trillion, a clot will form and can be detected. This is great news for us. Pretty much every single person who's ever had an injection of any sort has been protected because of this compound from this strange ancient creature. The only problem is that for this test to be readily available, pharmaceutical companies need a large supply of the blood of live crabs, which, as you'd guess, is not such great news for the crabs. In theory, the process of extracting blood from the horseshoe crabs doesn't kill them. It's sort of like blood donation, albeit a non-consensual one. And once their blood is taken, the crabs are released in a new location so they don't accidentally get caught a second time, ensuring they have a chance to recover. Their blood volume rebounds in about a week, and the LAL industry states that there are no long-term ill effects for the crabs. They measured mortality rates of less than 3%, but conservationists tell a different story. Between 10 and 30% of the bled animals, according to various estimates, actually die. And 30% of the animals per year dying equates to losses in the hundreds of thousands. And this isn't just bad for the horseshoe crab, but for the entire ecosystem in which they live. Many other species of animals rely on the horseshoe crab's eggs for food, like shorebirds and turtles. So the obvious question is, why haven't scientists made a synthetic alternative to LAL? Since the 1970s, they've certainly been trying, and luckily for the crabs, they have started to have some success. In 1995, scientists from the National University of Singapore were finally able to identify and isolate the gene responsible for the endotoxin-sensitive protein called factor C, the most important component in the LAL test, and produce it in yeast. Several years after that, they were able to create a rapid endotoxin test based on this recombinant protein. But despite these advances, these synthetic tests are still not widely available. They've been adopted extremely slowly due to regulatory and safety concerns. Europe didn't recognize the synthetic protein as an alternate endotoxin detection until 2015, and the FDA in the US didn't approve the first drug that used an endotoxin test based on the synthetic protein until 2018. And earlier this year, the American Pharmacopoeia, which sets the scientific standards for drugs and other products in the US, declined to place the synthetic protein on equal footing with crab lysate, claiming that its safety is still unproven. For now, we still need the horseshoe crab and their baby blue blood. But as more and more studies come out that demonstrate the safety of the synthetic version of the endotoxin test, the horseshoe crabs can breathe a bit of a sigh of relief. While they still face threats from overfishing for bait and habitat destruction, the adoption of this technology will relieve at least one major pressure. Our medical need for horseshoe crabs is what has started to push these animals towards extinction in recent decades. But this is not the first time they've faced such a profound threat. Since the first days of the horseshoe crab's ancestor, they have faced, and survived, all 5 mass extinctions. These extinction events are defined as a loss of at least 75% of species, happening in the geological blink of an eye. Volcanoes erupting, oceans warming, ice sheets forming, or oceans acidifying. The great die-offs result from a perfect storm of multiple calamities. The horseshoe crab and its ancestors were one of the few creatures to survive. But if so many things die, how does life rebound to flourish again? This is the question that researchers at the University of Oslo are trying to understand, and is the focus of the documentary Breakthrough: Recovering from Extinction on CuriosityStream. They are pioneering an investigation about what survived and what emerged after the largest mass extinction on our planet, 252 million years ago. This is one of many paleontology documentaries on CuriosityStream, which are all really good. And now, CuriosityStream has partnered with us to offer an incredible deal. By signing up to CuriosityStream, you now also get a subscription to Nebula. Nebula is the streaming platform made by me and several other educational YouTube content creators. It's a place where we can upload our videos ad-free and a place where we can experiment with new, original content. The original content is the best part of Nebula. Series like The Logistics of D-Day, or the game show Money made by Tom Scott, or Braincraft series Questionable Advice, series you can't get anywhere else. So by signing up at curiositystream.com/realscience, you'll get a subscription to CuriosityStream and a subscription to Nebula for just $14.79 for the entire year. Signing up is also the best way to support this channel and all of your favorite educational content creators. Thanks for watching, and if you'd like to see more from me, the links to my Instagram, Twitter, and Patreon are below.